Yesterday, as I explained you, this whole that cycle of glycolysis, fine, where by the breakdown of glucose, uh, we are getting two molecules of pyruvic acid or pyruvate, and whole this process does not require oxygen. This we have done yesterday. Now today we will start with the next part of this, that types of respiration. Means what happens to this pyruvic acid which is formed after the glycolysis. So there are three major ways in which different cells handle pyruvic acid produced by the glycolysis. So glycolysis के बाद ये pyruvic acid जो है वो थ्री वेज के थ्रू जो है आगे प्रोसेस में एंटर करता है. सो व्हाट आर दीज थ्री प्रोसेसेस? These three processes are the lactic acid fermentation, alcoholic fermentation, and the third is aerobic respiration. So students, one thing I want to tell you, that these both the processes are fermentation. First is the lactic acid fermentation and the second is the alcoholic fermentation. Both are fermentation taking place in different organisms. Like lactic acid fermentation takes place in the animals, and alcoholic fermentation it takes place in the fungus. So this is the difference here, but both are the anaerobic respiration. Then the aerobic respiration, as I said you, that aerobic respiration means here the pyruvic acid enters into the Krebs cycle, which requires the oxygen. Now first we will see the lactic acid fermentation. So let me revise here that the glucose is converted into the glyceraldehyde 3-phosphate, then 3-phosphoglyceric acid, then phosphoenol pyruvic acid is formed, which forms the pyruvic acid. This pyruvic acid is converted into the lactic acid, and this takes place in the muscles in anaerobic conditions. So students, do you know, when we do means a lot of physical exercise in while you may say that working of the muscles are more. Suppose we are doing exercise or any work, physical work जो भी हम करते हैं जिसमें muscles का जो है use ज़्यादा होता है. So in that case what happens, this pyruvic acid is converted into the lactic acid in order to provide the energy faster, because जितना ज्यादा वर्क होगा उतनी ज्यादा एनर्जी की जरूरत पड़ती है मसल्स को, तो जो मस्क्यूलर वर्क जब ज्यादा हो, in that case this lactic acid is formed in the muscle in anaerobic conditions, and we obtain some energy after this process, and this reaction is catalyzed by lactate dehydrogenase. So conversion of pyruvic acid into the lactic acid in the muscles in anaerobic condition, and here the enzyme which is involved, this enzyme is lactate dehydrogenase, and here NADH is actually NADH2, better to say, is reoxidized into the NAD. So this is the reaction taking place in the muscles. So this is taking place during the vigorous exercise when oxygen is inadequate for cellular respiration. So pyruvic acid is reduced to lactic acid by using the enzyme lactate dehydrogenase. Now we will see the next one, alcoholic fermentation. What happens in alcoholic fermentation? In fermentation, like by yeast, incomplete oxidation of glucose is achieved under anaerobic conditions. Again, anaerobic condition means no oxygen. This is taking place by the sets of reactions when pyruvic acid is converted into the carbon dioxide and ethanol. So I'll show you here that pyruvic acid is converted into ethanol. Pyruvic acid formed at the end of glycolysis is converted to the alcohol by using two enzymes. So here two enzymes are involved, pyruvic acid decarboxylase and alcohol dehydrogenase. यहाँ पर दो enzymes जो है वो utilize होते हैं to convert pyruvic acid in the ethanol. Here CO2 is the byproduct. So दो स्टेप के अंदर, फर्स्ट स्टेप के अंदर क्या होता है? इन द फर्स्ट स्टेप pyruvic acid is decarboxylated रिजल्टिंग इन द फॉर्मेशन ऑफ एसिटाल्डिहाइड एंड CO2. तो जो पहला प्रोडक्ट बनता है इसमें एसिटाल्डिहाइड बनता है, that is the decarboxylation of the pyruvic acid, hence the CO2 is also released. In the second step, acetaldehyde is reduced to alcohol. So final product we are getting alcohol or ethanol by 2 NADH here, and the product is ethanol as well as CO2. So this is alcoholic fermentation. So these enzymes, pyruvic acid decarboxylase, which is the first enzyme, and the second is dehydrogenase enzyme. Now the here the energy which is released, it is less, okay, less than 7% energy in the glucose. So ethyl alcohol does not means stay inside the microorganism, but is excreted out. And accumulation of alcohol beyond a certain limit kill the microorganisms also. As I said you कि fungus yeast के अंदर होता है, bacteria के अंदर भी ये कुछ certain bacteria are also there in which it takes place. तो ये जो है certain concentration तक ठीक है, अगर इसकी अल्कोहल की concentration अगर ज्यादा हो जाती है तो पॉइजन हो जाता है. Yeast poison to death when concentration of alcohol reaches about 13%. Okay. So therefore maximum concentration of alcohol in beverages that are naturally fermented is 13%, and a higher concentration of alcohol in a beverage is achieved through the distillation. So जो naturally जो beverages जो alcoholic drinks prepare होते हैं उनमें 13% ही होता है, बाकी जो है उसको distillation के through increase किया जाता है. So this is the alcoholic fermentation. Now we'll come to that special features first before I discuss कि difference क्या हुआ फिर aerobic and anaerobic में, क्योंकि जो fermentation है lactic acid fermentation and alcoholic fermentation is actually providing the energy, but both this fermentation they do not produce much energy. Less than 7% of the energy in glucose is released, and not all of it is trapped as high energy bond of ATP. Just now I that explain you, this is the difference. So energy release is less than 7% energy, मतलब ग्लूकोज की केवल 7% एनर्जी रिलीज होती है. Both the processes are hazardous, either acid or alcohol is produced. Net gain in both the types of fermentation is only 2 ATP. तो ये number of ATP भी जो है वो less produce होते हैं, only 2 ATP. And alcoholic fermentation results in the release of CO2 along with the ethanol, while lactic acid fermentation releases only lactic acid. तो जैसे मैंने अभी आपको lactic acid का fermentation बताया, यहाँ पे pyruvic acid का conversion, यहाँ पे CO2 release नहीं होती है, लेकिन pyruvic acid जब ethanol form करता है तब यहाँ पर CO2 release होती है. And the reducing agent NADH plus H plus, which is reoxidized to NAD plus in both the processes. So this reduction is taking place in both the processes. So understood, children? Now we will come to that type of respiration, that is aerobic respiration. So these previous processes were the part of anaerobic respiration. So what happens here? Here the complete oxidation of glucose takes place and energy extraction too. And in this process also the ATP molecules are synthesized. So it is the process by which organisms can carry out complete oxidation of glucose and extract the energy stored to synthesize a larger number of ATP molecules needed for the cellular metabolism. And it is common in higher organisms and takes place within the mitochondria. So as I explained you, the glycolysis is taking place in the cytoplasm of the cell. And this next in the aerobic respiration, after glycolysis, the next step, it takes place in the mitochondria. So this is taking place in the mitochondria. For that aerobic respiration process leads to a complete oxidation, as I said you. So here complete oxidation of organic substance and in the presence of oxygen, and releases CO2, water and large amount of energy present in the सबस्ट्रेट। तो ये की डिफरेंस हो गया, क्योंकि एनारोबिक रेस्पिरेशन में या फर्मेंटेशन में केवल 7% जो है ग्लूकोज का एनर्जी की फॉर्म में रिलीज होता है, यहाँ पर लार्ज अमाउंट ऑफ एनर्जी जो प्रेजेंट है सबस्ट्रेट में, दैट इज रिलीज्ड. And for aerobic respiration take place within the mitochondria, the pyruvic acid formed during the glycolysis is transported from the cytoplasm into the mitochondria. तो जो pyruvic acid cytoplasm में बना है, अब ये जो है ट्रांसपोर्ट होता है सेल के अंदर mitochondria में. And the crucial events which are taking place in it, let we discuss. So two main that events of the processes are there. The complete oxidation of pyruvate by the stepwise removal of all hydrogen atoms, leaving 3 molecules of CO2. And this takes place in the matrix of mitochondria. Matrix, you know, क्या होता है? Fluid होता है. So जो fluid है mitochondria का, उसके अंदर ये first step होगा. First event, complete oxidation of pyruvate by removal of all hydrogen atoms and leaving 3 molecules of CO2. Then the second event, the passing on the electrons removed as a part of hydrogen atoms to molecular oxygen with simultaneous synthesis of ATP. तो अब ये जो है electrons जो हैं वो remove हुए हैं as part of the hydrogen atoms, they are passed to the molecular oxygen, and at the same time synthesis of ATP takes place. Where does it take place in the cell? In the inner membrane of mitochondria. तो जो पहला वाला इवेंट है, स्टेप है, वो मैट्रिक्स में हो रहा है, और जो सेकेंड स्टेप है वो इनर मेम्ब्रेन में हो रहा है माइटोकॉन्ड्रिया की. ओके, नाउ हाउ डज इट टेक प्लेस हियर? So complete oxidation of pyruvate in the steps it is taking place. Again in this event 2 steps. तो जो पहले वाला है, जो कि मैंने आपको बताया कि पायरुवेट ऑक्सीडाइज हो रहा है और 3 मॉलिक्यूल्स कार्बन डाइऑक्साइड के रिलीज हो रहे हैं, और ये प्रोसेस हो रही है मैट्रिक्स ऑफ माइटोकॉन्ड्रिया में. अब इसके अंदर भी 2 स्टेप्स होती हैं. पहला, फॉर्मेशन ऑफ एसिटाइल कोएंजाइम ए. तो फर्स्ट होगा एसिटाइल कोएंजाइम ए का फॉर्मेशन, एंड द सेकेंड इज टीसीए साइकिल, दैट इज द क्रेब्स साइकिल और सिट्रिक एसिड साइकिल. फाइन. और जो सेकंड है ये होगा इलेक्ट्रॉन ट्रांसपोर्ट सिस्टम में, ये इलेक्ट्रॉन्स एंटर करेंगे और एटीपी जो है रिलीज करेंगे. तो हम पहले फर्स्ट वाले इवेंट को हम डिटेल में स्टडी करेंगे कि एसिटाइल कोएंजाइम का फॉर्मेशन कैसे होगा, और जो टीसीए साइकिल है या क्रेब्स साइकिल है या ट्राइकार्बोक्सिलिक एसिड साइकिल है, और वी कॉल इट सिट्रिक एसिड साइकिल ऑल्सो, हाउ डज इट टेक प्लेस. सो we'll start from the step that is formation of acetyl coenzyme A और ऑक्सीडेटिव डीकार्बोक्सिलेशन. ये दोनों नाम है इसके, कोई भी आप एक यूज कर सकते हैं. और यू अंडरस्टैंड कि फॉर्मेशन किसका हो रहा है? एसिटाइल कोएंजाइम ए का फॉर्मेशन हो रहा है. किससे? पायरुविक एसिड से. सो पायरुवेट विच इज फॉर्म्ड बाय द glycolytic catabolism of carbohydrate in the cytoplasm, after it enters mitochondrial matrix, undergoes oxidative decarboxylation by a complex set of reactions catalyzed by pyruvate dehydrogenase. So pyruvate dehydrogenase की मदद से ये oxidative decarboxylation होता है pyruvic acid का, and it requires enzymes also, like here you know the coenzyme A and NAD plus also. So these coenzymes are required. Magnesium also, magnesium ions are also required. Now after this process, what is formed? 2 molecules of pyruvic acid formed from one glucose molecule during glycolysis undergo the formation of 2 molecules of acetyl coenzyme A, 2 molecules of carbon dioxide, and 2 molecules of the NADH and the proton or H ions, H positive ions. And this reaction is called the link reaction or transition reaction also in the aerobic respiration. And the NADH2 molecule formed in this process enters the electron transport system of the mitochondria to release the energy. तो यहीं से जो है ATP का सिंथेसिस शुरू हो जाता है. ये जो NADH2 मॉलिक्यूल्स यहाँ पर फॉर्म हुए हैं ये एंटर कर जाते हैं ETS दैट इज इलेक्ट्रॉन ट्रांसपोर्ट सिस्टम के अंदर, एंड देन दे release the energy. So this is the first step. Understood? Formation of acetyl coenzyme A in the presence of coenzyme A and NAD plus. With the help of enzyme pyruvate dehydrogenase, magnesium ions are also required, and the formation of acetyl coenzyme A takes place. Now the next step is tricarboxylic acid cycle. So acetyl coenzyme A, which is formed here, it enters a cyclic pathway called as Krebs cycle, and Krebs cycle was named after the scientist Hans Krebs. So that is why it is termed as the Krebs cycle. So it is named after the scientist Hans Krebs, who first elucidated it in flight muscles of pigeon. तो pigeon के flight muscles में सबसे पहले Krebs जो scientist है उन्होंने पता लगाया था. And in this reactions, hydrogen atoms are removed from acetyl coenzyme A and transferred to the coenzyme further for further processing in the electron transport system. अच्छा, इसका दूसरा नाम है citric acid cycle also. So remember children, this everything you have to learn, that tricarboxylic acid cycle, that Krebs cycle, and the citric acid cycle, or we call it as CAC also. Okay, citric acid cycle. Why? Because of the formation of citric acid in the first step of this cycle. So this is known as citric acid cycle because the first that product here it formed during the reaction is citric acid. तो यहाँ पर citric acid का formation होता है. TCA cycle, that is tricarboxylic acid cycle, because many intermediate compound formed in the cycle have 3 carboxyl group. तो यहाँ पर जो intermediate maximum जो intermediate products यहाँ पर बन रहे हैं उनके अंदर 3 carboxyl groups होते हैं. That is why this is known as the tricarboxylic acid cycle. Understood children? Now the reaction of the Krebs cycle requires the presence of oxygen. This is also important. This requires presence of oxygen and are confined to mitochondrial matrix. तो ये कहाँ हो रहा है? Mitochondrial matrix के अंदर. And this cycle serves as a common oxidative pathway for carbohydrate, fats and protein. तीनों का जो metabolism होगा उसमें ये commonly होता है. All enzymes are soluble in mitochondrial matrix, but succinate dehydrogenase is found attached to the inner mitochondrial membrane. So let we come to the cycle. So it is the condensation of acetyl group with oxaloacetic acid and water. Citric acid in the presence of citrate synthetase. Okay, so सबसे पहले क्या बनेगा यहाँ पर जो है first product है citric acid. Fine. So here we will see that we have seen this formation of acetyl coenzyme from the pyruvate in the first step. Now this acetyl coenzyme A, further it forms the citric acid, and here it starts with the condensation of the acetyl group with the oxaloacetic acid and water to yield the citric acid. तो इसके अंदर और क्या involve है? Oxaloacetic acid and the acetyl coenzyme A. Fine. So condensation of the acetyl group, ये यहाँ से acetyl group condense होगा with oxaloacetic acid and water is also required to yield this citric acid, and this reaction is catalyzed by the citrate synthetase, and a molecule of CoA means coenzyme A is released. एक molecule coenzyme A का इसमें release हो जाएगा. एंजाइम जो यूज होगा वो होगा सिट्रेट सिंथेटेज. तो इस तरीके से सिट्रिक एसिड का फॉर्मेशन होता है. सेकेंड, सिट्रिक एसिड चेंजेस टू सिस एकोनिटिक एसिड बाय द एक्शन ऑफ एकोनिटेज. अब हम यहाँ पर आते हैं क्योंकि ये पूरा शॉर्ट में दिया हुआ है. डायरेक्टली यहाँ पर अल्फा कीटोग्लूटरिक एसिड, सक्सिनिक एसिड, मैलिक एसिड, and again this is regenerated oxaloacetic acid. I will explain you the cycle here in detail. So citrate here, or the citric acid changes to cis aconitic acid by the action of aconitase and here the water is released, and cis aconitic acid changes to isocitrate further with the help of the same enzyme aconitase isocitrate में convert होगा, and in this manner citrate is isomerized to form isocitrate. Now isocitrate is changed into that oxalosuccinate, and one molecule of NADH also produce this oxalosuccinate. अब यहाँ पर एक और स्टेप है जो यहाँ पर शो नहीं किए, डोंट वरी, आप आइसोसिट्रेट से सीधे अल्फा कीटोग्लूटेरिक एसिड भी बता सकते हैं. तो ये आइसोसिट्रेट में के बीच में एक और इंटरमीडिएट प्रोडक्ट बनता है ऑक्जेलोसक्सिनेट, और ये ऑक्जेलोसक्सिनेट फर्दर कन्वर्ट हो जाता है अल्फा कीटोग्लूटेरिक एसिड के अंदर. यहाँ पर कार्बन डाइऑक्साइड रिलीज होती है और एंजाइम जो है वो आइसोसिट्रेट डिहाइड्रोजिनेज, ओके, एंड दैट रिडक्शन ऑफ NAD to form NADH2. Now alpha ketogluteric dehydrogenase complex sets on alpha ketogluteric acid, and this is converted into 4 carbon compound that is succinyl coenzyme A. ये 5 कार्बन कंपाउंड है, ये सारे 6 कार्बन कंपाउंड थे. ओके, तो यहाँ पर 6 कार्बन से 5 कार्बन बना, एक कार्बन यहाँ पर कार्बन डाइऑक्साइड की फॉर्म में यहाँ पे रिलीज हो गया. अब ये है 5 कार्बन कंपाउंड, इससे जो फर्दर बनता है वो बनता है 4 कार्बन कंपाउंड. सो सक्सिनिल कोएंजाइम A is the 4 carbon compound, and this reaction is catalyzed by alpha ketoglutarate dehydrogenase. इसकी प्रेजेंस में होता है. Okay, here again one molecule of CO2 is evolved here, released, and NAD plus is reduced to NADH2. Fine, the same here taking place. Now further in the next step, this succinyl that coenzyme A further it is converted into this further converted into this succinate, or you can say this here succinic acid also, or succinyl succinic acid also, और succinate दोनों आप लिख सकते हो यहाँ पर. तो succinic acid में convert होता है in the presence of enzyme succinate thiokinase. Okay, so और succinyl coenzyme A synthetase B, दोनों एंजाइम के नाम करेक्ट है. So succinyl coenzyme A is converted into succinic acid or succinate in the presence of succinyl coenzyme A synthetase, and here you will see the formation of GTP. ATP you know adenosine triphosphate, और ये क्या होगा, GTP is the guanosine triphosphate. So here formation of GTP takes place in this process, and this is the substrate level phosphorylation. In a coupled reaction, GTP is converted to GDP with the simultaneous synthesis of ATP from ADP. तो इसी GTP से ATP का synthesis होता है, ये GTP जो है GDP में convert होता है, and then simultaneously the formation of ATP takes place. तो ये substrate level का phosphorylation है. Now succinic acid का formation हुआ. Now next step में fumaric acid or fumarate is formed from the succinic acid, and this reaction is catalyzed by the succinate dehydrogenase. So it is easy to learn कि जो भी यहाँ पर name है substrate का, on that basis dehydrogenase means this is the removal of the hydrogen. So succinic dehydrogenase is the enzyme which converts succinic acid into the fumaric acid or fumarate. Here again the FAD molecule is reduced to FADH2. Fine. Now further the formation of malate or malic acid from the fumaric acid in the presence of enzyme fumarase. And here what happens, this steps involves the addition of water. So ये जो formation हो रहा है malic acid का fumaric acid से, यहाँ पर water की requirement है. So it is involves the addition of the water molecule. Now further the regeneration of oxalosuccinic acid, because as I explained you कि oxaloacetic acid and here the coenzyme A together they form the citrate. Fine. So this oxaloacetic acid is regenerated, and the last step catalyzed by the malic dehydrogenase enzyme and reduces NAD plus to the NADH2 by removing hydrogen from the malic acid. जहां भी ये dehydrogenase अगर term आ रही, it means it is removing hydrogen from the substrate. Here also you can see isocitrate dehydrogenase. So यहाँ पर isocitric acid से जब अल्फा कीटोग्लूटेरिक एसिड बन रहा है तो हाइड्रोजन रिमूव हो रहा है. That is why NAD plus is reduced to NADH2. Everywhere you will see this. Succinic dehydrogenase is there where FAD is reduced to form FADH2. Here also NAD is reduced to form NADH2. So formation of oxalosuccinic acid from the malic acid. So understood, children? This is the whole process of the that Krebs cycle. These all are the steps involved. And here one more thing I will discuss with you, that one molecule of ATP via direct GTP and three of molecules of NADH2. So you can see one molecule here from when the alpha ketoglutaric acid is from isocitrate, second NADH2 here alpha ketoglutaric acid to the succinyl coenzyme A, and the third is from malic to oxalosuccinic acid. तो यहाँ पर 3 places पर NADH2 का formation हो रहा है. At one place FADH2 from जब succinic acid से fumaric acid बन रहा है, and 2 molecules of CO2 are released. You can see one is here and another is here. 2 molecules of CO2 are released. Per molecule of acetyl coenzyme A oxidized. मतलब एक मॉलिक्यूल जो एसिटाइल कोएंजाइम ए का ऑक्सीडाइज हो रहा है तो ये उसके प्रोडक्ट हैं. अब क्या हो रहा है? We know that 2 molecules of pyruvic acids are formed from one glucose molecule. So TCA cycle must occur twice for each molecule of glucose respired. तो 2 molecule है pyruvic acid के, तो ये जो TCA है ये twice होगा for each molecule of glucose. So नंबर अब क्या है? डबल हो जाएंगे. कैसे? 2 ATP. Okay, this is from the GTP itself. And 2 ATP, then 6 NADH2, क्योंकि 1 स्टेप में 3 बन रहे हैं तो क्या हो जाएगा? Here 3 2 6, twice डबल कर देंगे हम, तो 6 molecules of NADH2, 2 molecules of FADH2, they are formed by the 2 molecules of acetyl coenzyme A. And the continued oxidation of acetyl coenzyme A via TCA cycle requires the continued replenishment of oxaloacetic acid. ये बराबर जो है replenish होता रहता है, regenerate होता रहता है प्रोसेस के अंदर. And the first member of this cycle, and it also requires the regeneration of NAD plus FAD plus from NADH and FADH2. क्योंकि ये रिएक्शन जब लगातार हो रही है तो यहाँ पर NAD प्लस और FAD प्लस की भी कंटिन्यूस रिक्वायरमेंट बनी रहती है. सो इसकी जो समरी है वो हम किस तरीके से करेंगे, आप रिएक्शन पूरी इसमें लिख सकते हैं. पायरुविक एसिड इन्वॉल्व है, 1 मॉलिक्यूल, देन यहाँ पर 4 NAD प्लस प्लस FAD प्लस 4 H2O, यू नो वाटर की भी रिक्वायरमेंट है, 4 H2O प्लस ADP plus phosphate ion. So it releases 3 carbon dioxide, 4 NADH, 4 hydrogen ions, FADH2 plus ATP plus H2O. So इस तरीके से जो है ये जो वाटर एंटर करता है ये वापस से रिलीज हो जाता है. ओके, प्रोवाइडिंग द प्रोटोन. अब ये जितने भी प्रोडक्ट्स बने NADH2 and FADH2, they enter into the mitochondrial electron transport chain and release the energy in the form of ATP. So students,